Dear students, in this particular video, we will study about the bacterial conjugation process, which was discovered in the year of 1946 by Lederberg and Tatum. The process may take place in between a donor and a recipient cell, and the donor cell may be F plus or HFR, and the recipient cell always it is F minus. Okay, now we have to know what is F plus cell and what is F minus cell, or what is HFR cell. Suppose this one is a bacteria, it is its double-stranded circular DNA, and if any extra circular DNA is present inside the body and which is far away from that particular genophore, this one is genophore, and this circular DNA, extra DNA, so if it is present far away from the genophore, then this is termed as F plus bacteria, because this particular DNA contains F factors. Similarly, if it is having only that particular genophore and no extracellular DNA is there, then it is F minus. And if the bacteria is having the F factor attached to the genophore, then it is termed as HFR. Okay. That is, in F minus cell there is no plasmid in the cytoplasm, only genophore is present here. That is, only genophore is present. This one is genophore, double-stranded DNA. No other extra plasmid is there. Okay. And in F plus cell, the plasmid is located in the cytoplasm along with the genophore, but there is no direct contact in between them. That is, if this one is the genophore, that the plasmid is far away from the genophore. Okay? And in case of HFR cell, it is clearly written that the plasmid is in direct contact with the genophore as episome. That is, it will be like this. Genophore and plasmid are attached to each other. Clear? Okay. Now, by the diagram, we will study about one F plus or donor Escherichia coli cell. Clearly, the diagram is showing you the genophore and the F plasmid, which are far away from each other. It is to be noted that the genophore is also double-stranded DNA, as well as F plasmid is also made up of double-stranded DNA. And due to the presence of F plasmid, sex pilus or sex pili are appeared from the surface of the bacteria, and their number is minimum 1, maximum 5. Clear? In this diagram, only double-stranded circular or genophore, that is, the nucleoid is present in the bacteria. No other extra DNA or plasmid is present in the cell. and there is no any sex pilus also. Such a cell is termed as F minus or recipient because it receives the genetic material from the donor. Clear? Okay. How it will be converted into F plus? If any factor, F factor, is inserted inside this one and it is kept far away from that one, then this F minus will be converted into F plus. And this conversion is the main motto of this conjugation process. That is, we may say that by means of conjugation process, one F minus recipient Escherichia coli is converted to F plus by receiving the genetic material of F plasmid from the donor cell. The donor may be F plus or donor may be HFR. Now we will study the process of conjugation in between F plus and F minus cell. Look at the diagram. Here two bacteria are in contact to each other. One is F plus, another one is F minus. Why it is F plus? Because due to the presence of this particular plasmid. And why it is F minus? Because there is no any F plasmid. Okay. And due to the presence of F plasmid, the bacteria present in left hand side, that is F plus, is having few sex pilus on the surface, but this one is without any sex pilus. Okay. So at the beginning, this F plus will made a contact with the F minus by means of sex pilus. That is, the bacteria, this bacteria will made a contact to this one by this sex pilus. Clear? After the formation of the contact or breeze, this particular sex pilus will be retracted or it will be pushed back inside the F plus bacteria. Clear? And due to this activity, the F minus cell will go towards the F plus cell. Again I am telling, this particular sex pili of the F plus cell will be retracted within the F plus cell. That is why this F minus cell will go towards the F plus cell, and as a result of that, the distance between these two cells will be reduced. Clear? Again I am telling, after the formation of the contact, the F plus cell will pull the F minus cell towards itself by means of the contacting pili. Clear? Okay. After that, what happened? After that, there will be a change in the F plasmid of the F plus cell. What is that? Here origin of replication is present. That is, if this one is the double-stranded circular F plasmid, then in a particular zone origin of replication is present, and at that origin of replication, rolling circular mechanism will be performed by the formation of a nick, and due to that process, one strand out of the two strands, one strand of the F plasmid DNA will be broken down at a particular point, and it will be converted into a linear thread. Okay, here by this diagram it is clearly shown that this particular outer strand has been broken down at a particular point. Okay, and in that particular point the nicking enzyme is present, and this nicking enzyme will push the strand towards the bridge, and it will pass to the F- cell through the bridge. Clear? Again I am telling you that what happened if this one is the F plus cell and this one is the F minus cell, and this is the point of contact or the bridge or the tunnel, then out of these two particular DNA strands, one will be broken down at a particular point. As a result of that, the DNA will be like this. This one is 3 prime and this one is 5 prime. Okay, the nicking enzyme will push the DNA, that is broken down DNA, to the bridge or tunnel, and it will push this particular DNA to the recipient cell. Clear? Okay. Now, due to the transfer of the DNA strand, single-stranded DNA strand, the F minus will get a strand from the F plus cell, and F plus cell's plasmid will itself be in a single-stranded condition. That is, we may say that the F plus DNA's double-stranded structure will be converted into single-stranded, and one strand will be transferred to the F minus cell. Clear? That is, one single strand will be remain in the F plus itself and another one will be transferred to the F minus cell. Okay. Now both of these cells are having genes or factors inside them, but both of them is having single-stranded DNA in them. Okay, as plasmid, there is no change in their genophore. Only the plasmid of F plus cell has been converted to a single-stranded one, and here it is clearly indicated that this cell will have only the lagging strand and its leading strand has been transferred to the F minus cell. We know that the plasmid DNA is double stranded, but in both the cells the plasmid DNA is now single stranded. So DNA replication will take place, and by means of DNA replication both of these single stranded DNA in the plus cell and minus cell will be converted into double stranded DNA. Thus the F plus cell will regain its original condition and F minus cell will be treated as F plus cell because of the presence of this double stranded DNA ring. Okay, and we have seen that this was from the F plus cell by means of the conjugation process. After the formation of such new double stranded DNA, that is the plasmid having one old strand and one new synthesized strand, both of the F plus and F minus cell will be separated from each other. That is, the tunnel or the point of contact will be broken down and they will be separated from each other. Clear? So we have seen that the F plus again regain its original condition, that is it will have again DS, that is double stranded F plasmid DNA, and the F minus cell in the right hand side, previously it was written as F minus, now here it is F plus. Why? Because of the presence of this particular structure. That is, we may say that by the conjugation process the F minus cell has been converted to F plus cell and there is no any change in the F plus cell. That is, F plus cells remains the same whereas the F minus cell has been converted to F plus. Okay, now this cell, this particular cell, it will behave as a donor cell. That is, we may say that by the process of bacterial conjugation process or by this method, we may say that the F minus cell has been converted to F plus cell. Okay, so this is about the bacterial conjugation process. Okay, thank you.